the content of the meeting. As Amelia just said, I'll just outline what is going to go on, and I'm going to give a short introduction to why we have this focus on deep rooting for this meeting. Then we had planned a keynote presentation from Matthias Visuva, who has worked a lot with root traits over many years, but unfortunately he's not able to make it today. But luckily then Philippe Insinger will tell us some of the same stuff and talk about some of Matthias' work and some other aspects of why root traits are important. After that, we'll have 3 short presentations of various aspects of the SolACE project work. And we will have, now that the meeting is online and we cannot be together, we will have made a virtual tour of our root phenotyping platform in Copenhagen that will be shown. And then there will be time for further questions and discussion. So why all this focus on deep rooting? I mean, the SolACE project deals with crop resource efficiency. We need to improve that in future farming, and also we're sure we need to improve crop resilience as global climate change is continuing and making more unstable and often more dry conditions. We need crops that can still produce under those conditions and continue producing when the water supply is low and when maybe nutrient fertilization is regulated to avoid environmental problems. And the focus on deep roots, of course, when we want better resource efficiency, roots is an obvious thing. We want the crops to be better to assimilate the resources from the soil. And especially when we look at water and nitrogen, they are mobile in the soil, meaning on one hand that they are relatively easy for the roots to take up, but on the other hand that as soon as there's too much of either of them, they will tend to move deeper into the soil. And for the cropping system to be efficient, it has to have deep roots and be able to recapture those resources. It's most well understood for water, where we typically have a winter period with surplus precipitation. This water will move down into the soils, and unless we can access that during summer, we will experience severe drought and yield loss. It's just illustrated simply in this graph. I can illustrated here, where you have one cropping season. The soil, you can see the gray part here is the soil drying out, and we come into a winter season. The soil is being filled up with water again, and into a new season where early on the crop have shallow roots and take the water from the upper soil layers, but as time goes on, the crop roots grow deeper and can continue to take water from deeper into the soil. So that is basically a very simple concept, but requires a lot of the root system of the plant. It has been shown in some experiments that this ability to acquire deep water late in the season can have a very big yield effect. To understand how we look at the crop root zone, you can see here an idealized image for how a wheat root system could look. You can say that in the upper part of the soil, and for a winter wheat that could be like the first meter, there will typically in the full developed crop be sufficient roots to take up whatever water and nitrogen is there. If they don't take everything, it's due to other limitations than the roots. Then we have some zone below that, could be somewhere between 1 and 2 meters, where we have roots developing. They come late in the season, they don't reach the same high density, but they still take up quite a bit of resources. So what we see is you have this more or less full resource use in the upper soil layer, then declining resource use in the next soil layer. And what, of course, we want to do by breeding is to move this border zone downwards. So maybe if we are successfully in a breeding cycle, we can add 10, 20 centimeters of root depth, and we can add the resources in this soil layer to the crop for growth and production. This is just an illustration of how we have seen a wheat root system develop in the soil over time. You can see starting in November in the winter wheat, few roots, and only in the topsoil. Then over the late autumn into December and March, still few roots, but quite a bit of root growth over winter. But then we have until May and until June, we have the really strong root growth, and we have roots developing down to 1.5 and 2 meters and even a bit more. And then in the grain filling part, the root growth is slow, but we still often see some root growth in the deep part of the soil profile. So I think to quite a lot of people, it's a bit surprising to see that these crops actually grow and use water and nitrogen from such depths. Here it's a graph showing for different from some of our earlier work for different genotypes, how they developed over time in terms of root depth in 2 years. And of course, there's not a huge difference. If you look at the end of the season, you can see that there is some difference between them in the order of 10, 20, 30 centimeters. And that may not sound like much, but still, the amount of resources in such a layer, if that is added to the crop resource base, that can be quite a lot in a stressful situation. We measured the soil nitrate at harvest in these experiments and can see that down in the deeper layers, actually we see quite a lot of difference between here, depending on the full lines are at a high nitrogen levels, the broken lines at a lower, but we see that at the end of the season also deep in the profile. And if you look at the green curves, this one, and also here, that's the genotype that had the deepest roots and also reflects into less nitrogen left in the subsoil at the end of the growing season. We are working with this in many ways, and you will see later on various, this is a picture of the facility where you will get a virtual tour a little bit later. So one of the things we are doing now is that we are working together with breeders to look for the genetic variability in deep rooting, and a facility like that, we get data like this. You can see here the names of a lot of genotypes and breeding lines, and you can see the very different here recorded as the root length we saw below the depth of 130 centimeters. And one of the things we also focus on, and this is an early attempt, is of course we want this to translate out into real fields and real yield advantages and real advantages to the breeders. And in 2018, in our part of Europe, we had a very dry season. I think that was quite widespread. And Simon Fiel-Svane, who was working on the project at the time, took root data from this facility and compared them to the genotypes that were also out in farmers' field tests of comparing genotypes, and compared our root measurements in the Radimax facility to the yield on these farmers' field trials of genotypes. And as you can see here, we got quite a nice correlation that indicates that what we measure in this facility can help us improve resource efficiency and yield. So with that, I hope I have set the scene for why we are so focused on the deep rooting, and you will hear more both about this facility that we have at the University of Copenhagen and other results from the SolACE project in the next minutes. So with that, I would like to start the meeting program by handing over the phone and screen to Philippe. So please, Philippe, let us have your presentation. You are muted, Philippe.
Sorry for that. Thanks, Christian. I'll try again. So I do hope you see my screen. I'm going to talk about root traits for efficient resource acquisition. Mostly I will focus in the examples I have on nutrients, and a lot of them are on phosphorus, because this is my own field of research. I work at INRAE in Montpellier in south of France, and I'm happy to be the coordinator of SolACE. So as Christian was explaining, Matthias Visuva from JIRCAS was supposed to talk, couldn't make it, unfortunately, today. So I'll try my best to replace and show some of his beautiful work on root traits. First, one thing I would like to stress is a paper that was published in Nature 12 years ago, pointing an underground revolution, and that plant breeders were turning their attention to roots. In this paper, there was some interviews, and especially one from Jonathan Lynch about his paper, The Roots of the Second Green Revolution. Jonathan Lynch is going to be keynote speaker in the next event of this sort, of this series, on the 10th of February, by the way. And I've taken a few things, ideas from his paper. He was pointing that the green revolution was based on crops responsive to optimal conditions, and that the second green revolution shall be based on crops coping with suboptimal conditions, for instance, low soil fertility or drought. And this means, well, he's a root specialist. This means that we should look for crops that are exhibiting improved resource acquisition efficiency through either both or both foraging strategy or mining strategy of roots. I mean, most of my talk is going to be about foraging strategy, and just one slide for the mining strategy. What does it mean? I mean, foraging means traits that are involved in increasing the volume of soil explored, as stressed already by Christian, or the rhizosphere volume, if you prefer, while mining strategy is about increasing the resource availability in this soil volume affected by roots, which is called the rhizosphere. So I've got to start with this great example of a work by a geneticist, Matthias Visuva, started in the early beginning of this century. And that's a work where he looked in very poor soils, that was in Philippines, volcanic soils, high phosphorus fixation capacity, so a very big constraint on phosphorus availability. So what he did was comparing the most commonly used variety of rice, Nippon Bear, which was not very efficient. You see, or you almost see on this slide, on the picture, that it has no grain actually in these kind of conditions. Whereas compared to it, they came up with a near isogenic line called PUP1 for phosphorus uptake that produced some yield, as you can see. And actually this was related to a much greater efficiency for phosphorus acquisition or uptake. You see on the table that actually the uptake was increased by 200% compared with the inefficient variety. And what is interesting in his work is that he tried, based on number of measurements and then on modeling, to explain this kind of huge increase in efficiency in uptake. And you see here that looking at first at a quite simple trait, which is specific root surface area, so the surface per unit of root biomass, that only 20% increase of this trait was enough to explain this 200% increase of phosphorus uptake. In his calculation, he also showed that another trait, which is more related to the mining strategy now, which he called external P uptake efficiency. This is the amount of phosphorus that is susceptible to be taken up per unit of surface area of root. And he showed that only 30% of increase of this trait was enough. So that's why he called his early paper small causes with big effects. Maybe it's a nightmare for a geneticist, because looking for such little changes in a root trait is not that easy, but it was good news. There was no need to increase by 200% the root size to get this kind of impact on the performance of the crop. A few years later, they published this paper where they actually identified the gene involved in this efficiency, and they call it PISTOL1 for phosphorus starvation tolerance. What was interesting in this work they published was that they realized that most of the modern germplasm did not have this gene, so suggesting possibly that it had been counter-selected. While when exploring traditional germplasm, usually this gene was found. And they also showed that this gene was actually involved in root growth regulation. I'll come back at the end of my talk with more recent work from his group. In addition to this, there are a number of other examples I would like to show, and in different conditions, in European conditions and crops, important crops. So this is an example in our group, in a former European project where we look at root traits in maize, and using the approach popularized by Jonathan Lynch called shovelnomics. We looked at traits of the topsoil and showed here that there was a decent relationship between phosphorus acquisition among these 23 genotypes in 2 different soils with the root surface area. It's not the only trait I've been already mentioning several times. Another very efficient way to increase the resource acquisition through increasing root surface area is through root hairs. And this is an example of old work by people in Denmark on barley genotypes, showing variation in root hair length, and the consequence for huge consequence for variation in soil volume that is explored. And their work showed also in the field that there was some distinct increase in phosphorus acquisition that was likely to be related to root hair length. So that's another trait that is worth pursuing. Well, as was pointed already by Christian in his introduction, we tend to look, because that's easier, to look only at roots in the topsoils while crops grow roots pretty deep in the soil. This is famous drawings from Kuchera for wheat. And you see that even in quite early stages, there is significant roots below the topsoil. And obviously towards the end of the cycle, it's even more important, although most of our work is on topsoil roots. Another example here from sugarcane, so it's getting back to the tropics, but showing that in some crops, we are talking about really deep roots. You see on the scale, it's down to 4 to 5 meters depth. And what is also interesting in this representation of root densities through depth is that there is an even greater density of roots when plants are experiencing limitations, like in this case, drought. So in the rain-fed conditions, you see much more roots at depth and in the subsoil, and that's showing, if you don't know, that roots are very plastic, which is interesting, but also complex then to study. On top of these root traits, we shall not forget that plants are also relying on other important participants in resource acquisition, and some of them are Mycorrhizae. So this is another example from our previous work on maize genotypes in field conditions at 2 levels of phosphorus. With we looked at 2 different species of mycorrhizal fungi in the roots. And you see first that there is, if you look at the various points, that there is considerable variation among these 23 genotypes. And also that there is a differential response to phosphorus availability. Usually in textbooks, we hear that phosphorus fertilization is decreasing mycorrhizal colonization of roots. This is what you see with the top case of Rhizophagus irregularis, but we just observe the opposites actually for the other species of mycorrhizal fungus. So we definitely need to further look at mycorrhizal symbiosis and these kind of traits, because through their external hyphae, mycorrhizal fungi contribute significantly, usually, to exploring a greater soil volume and therefore more resources in the soil. This is my example and single example, although that's my favorite activity when I was a scientist. That's about the mining strategy of fruits. It's a whole range of processes involved in the rhizosphere to, in this case, increase the availability of nutrients, of possibly water, through a number of different processes. This is an example here with phosphatase activity. Phosphatase are enzymes that are used by all sorts of organisms, bacteria, fungi, plants, to increase the availability of phosphorus through mineralization of organic phosphorus in soils. And what you see in this plot for 2 different soils, an alkaline soil, neutral soil, 2 levels of phosphorus, that's usually what we observe, but again with considerable variation among the 23 genotypes of maize. We usually see an increase of phosphatase activity in the rhizosphere, in the volume of soil just around the roots compared to the bulk of the soil. So this means that it's another trait, an example of a trait involved in mining nutrients that we should also look at when we want to better understand acquisition efficiency in crops. And finally, I would like to point that roots are not living on their own. We've seen that already when talking about mycorrhiza, but beyond mycorrhiza, plants live with a complex microbiome as any other living organism in the planet. And this is an illustration here from a review paper by Laurent Philippot. By the way, Laurent Philippot is going to be the keynote speaker at the 3rd event of this series on the 3rd of March. And this is showing the diversity and the change in the composition of the microbial community in 3 different genotypes of maize from the 23 I showed before, and also a big difference between them and a big difference with another plant species, which is sugar beet. So again, this means that crops differ in the microbial community they harbor in the rhizosphere. And that means that there are opportunities to take advantage of this. So to conclude, as was pointed in this review paper many years ago now, we have 2 strategies that we could use to try to improve acquisition of resources and stress tolerance, either through root plant strategies and looking at root traits, or through the microbiome, so microbial strategies. I won't go further into the details of this slide, but that's an important thing. And to end up with, I'm coming back to this success story of Matthias Visuva with very recent results that he showed at the conference at the end of last year. So what they actually did with their PUP1 genotypes is that they make some backcross lines with derived from IR64, which is very common genotypes variety used, and especially very widely used in Madagascar. They did this backcross and then they send these lines to Madagascar to complete the selection process in farmers' field with local conditions, very little use of fertilizers, and actually zero input in this selection process under varying native soil fertility conditions with farmers. And you see that the process went from 2016 to 2021. And these are some of the results they obtained with 2 lines that are now released as varieties. So 5-vari32 and 5-vari85 that are outperforming, in this case, IR64, and also a local variety that is largely used by farmers. So we hope that this is going to be taken up, these novel varieties that are very nice examples of success story when looking at root traits and selecting for more efficient root traits for resource acquisition efficiency in crops. So with this very last example, I would like to finish and come back to these many promising subterranean strategies we are looking at to face global change, especially climate change, and achieve an agroecological transition of our agriculture. As I pointed several times, this is not just about topsoil, young roots, and not just about roots. And in the SolACE project, we've been trying to look deeper. And so we will have some examples now to look at what's happening deeper in the soil. So thanks a lot. And maybe if we still have time, I can answer some questions.
Yes. Are there any questions? We don't have much time, but one or two questions maybe.
Feel free to, since we didn't get any questions in the chat, you're welcome to pop them in the chat quickly and we'll read them aloud, or you can unmute yourself and ask a question out loud, since we do have a couple minutes. So here's the first question from Pierre. How relevant is shovelnomics for wheat root characterization?
Oh, that's a good point. Shovelnomics, I didn't explain, is just using a shovel in the field to extract the roots, and that's obviously easier from the topsoil than from the subsoil. The nice thing about shovelnomics is that you can do it at any growth stage and looking at real roots in real soils. Problem is that it's very tedious to get the roots of the soil and to avoid breaking them too much, to collect the whole of them. And obviously, the finest the root system, the more difficult it is. So that's why I showed the example with maize, because maize has got quite coarse roots compared to many other crops. This is obviously much more difficult with wheat and even more with some other crops. And indeed, it's a challenge. So, but still, you can it can provide some examples, some data that are complementary to data that you can get through other approaches. And in this morning and in the next event, there will be some example of different types of phenotyping strategies that we can use, applying them to wheat. One of the things that is difficult for wheat is looking at angles, like what we did in maize is obviously slightly more difficult with the shovelnomics approach, again, because the roots are finer. But it's possible.
We have another question from Cecile who asks, have you characterized the microbiome composition of the 23 maize genotypes in your example?
No, that was a work that was conducted a few years ago, and we actually focused on Mycorrhizae. And even for Mycorrhizae, we didn't look at the whole of the community. We used a qPCR approach at that time, and we only looked at 5 species.
Great. Thank you very much. And just to reassure everyone, if you have more questions, we will have more time for questions later on, but we're going to move on. Thank you, Philippe.
Thanks.
Yes, thank you, Philippe. And then the next speaker will be Anesta O'Donnell, who will talk about some of the work we have done at Copenhagen University on it. Please, yes.
Yes, hello, good morning. My name is Anesta O'Donnell. I'm a PhD student at Copenhagen University, and I will show you briefly some results from the Radimax facility in Denmark. So first of all, this is the Radimax facility. You can see it down here on the left. It's a root screening facility. And in this experiment, we had 14 genotypes of winter wheat in 2019 and 2020. And we were looking at combined water and nitrogen stresses. So down here on the right, you can see a cross-section of the facility. And we've imposed a water stress gradient within the row. So you can see on the sides there's less soil and therefore less water available to the plant roots. And in the center there's more soil and more water. So that's where we get the wet treatment. And then we also had 2 nitrogen treatments, high and low, of 190 and 60 kilos per hectare of nitrogen. And those are applied to the soil surface for the separate rows. We had 4 root imaging campaigns for each year. So this is our wagon with cameras on it up here on the top right. And you can see the small tubes that the cameras go into. And these run along the surface of along the bottom of the bed. So we can take images of the plant roots. And finally, we have some irrigation tubes which run horizontally along the bed. And at anthesis, we injected a nitrogen and a water tracer at 180 centimeters, so into the deep soil, so that we could see how much of the nitrogen and the water was actively being taken up by the deep roots. So firstly, to show you some of the roots, these are root profiles from in the soil in 2019 and 2020. So you can see we have the soil depth going down to 2.5 meters. And this is the average root length at each of those soil depths. So in green, you can see 2019, and in blue is 2020. And I've just highlighted the June data from each of those years, because those give us the best results that we most frequently use. And you can see that we do have roots going down to around 2 meters in both years. Although there are a lot more roots in 2019 than in 2020 for various reasons. Here I've just divided these, on the left, I've divided these results up into the high nitrogen and the low nitrogen treatment. And again, you can see that there are a lot less roots in 2020 than in 2019. But we also see here that there's not such a huge difference in the root lengths between the high and the low nitrogen treatment, although there does seem to be slightly more in the low nitrogen treatment. But of course, we really want to focus on the deep roots. So here on the right, this is a plot of a trait that we've developed to try to measure the deep rooting. And in effect, it's a measure of the soil depth below which we have 40 centimeters of roots. So you can see again in 2019 there was a lot more root than in 2020, and it does seem to be slightly more root in the deep root in the low nitrogen treatment, although it's not significantly different here. So now turning to the above ground measurements. On the left here we have the grain yield for the 2 years, and you can see the blue lines show the high nitrogen treatment and the green lines show the low nitrogen treatment. Well, the dark is the wet treatment and the light colors show the dry treatment. And we can see that in both 2019 and 2020 there was a strong effect of both of the treatments. And in particular, nitrogen had a strong effect. On the right, you can see again 2019 and 2020 the harvest index. And there is in particular an effect of the water treatments, although not such a strong effect of the nitrogen. And I've divided these here up into the different genotypes, and of course that's a lot to look at. So I've just selected a few highlights of them here for you to see some of the ones that I thought were interesting. So on the right here we have the CH73641. You can see there's not a huge difference in the grain yield between the 4 different nitrogen and water treatments. Whereas in the middle we have the pageant, which has quite a strongly been affected in the grain by the water treatments, while on the left the creator has been affected quite strongly by nitrogen, but not so much by water. And below I've just plotted the root profiles, so that, sorry, the root profiles, so that you can see what they look like. And the idea here is that we want to see how these above ground and below ground traits interact, and how the roots can affect how the nitrogen and water treatments affect the yield. So, oops. Yeah, so combining the grain yield and the root length, here I've plotted the grain yield versus this deep root trait measurement for 2019 and for 2020. And the red is showing the high nitrogen treatment and the blue is showing the low nitrogen treatment. And we have 2 lines for each because we have 2 separate beds or blocks within the facility. But we can see in all of these cases that we have a positive relationship between grain yield and roots, which means that the more there are roots in the deep soil, the higher the grain yields are generally. And there does seem to be a stronger relationship here between the high nitrogen treatment and than the low nitrogen treatment. Next, I will just show you this is from the nitrogen tracer that we injected into the deep soil. So here we can see the different genetic material and how much of this nitrogen tracer they took up from the deep soil layer. And we can see there are definitely some big differences between the genetic material, although there is a lot of variability. And this is due to a number of factors, but probably mainly because there are some big differences between the 2 beds or blocks within Radimax. And we do see some spatial variation there. But here I've just plotted the same measurement, how much of the nitrogen tracer has been taken up against this deep root measurement. And we do see, particularly in the bed 2 here, both of these results are from 2019. But we do see, particularly in the bed 2, that there is a strong relationship between how deep the roots are and how much of the nitrogen they take up from the deep soil layer. And this is higher in the low nitrogen treatment here in blue, which may be related to the slightly deeper roots in the low nitrogen treatment. Next, I'll show you this 13C measurement. And this is a measure of a natural process which indicates water stress. These are measurements from the grain. So again, we can see some big differences between the different genotypes and how much water stress they experience. And the taller bars here show less stress, less water stress, and the smaller bars show more water stress. And here I've just divided those again into the high nitrogen treatment in red and the low nitrogen treatment in blue. And you can see that overall, or generally, it seems like the low nitrogen treatment actually has less stress, which is what we would expect, because the low nitrogen treatment generally have less growth and therefore less demand for water. And here I've plotted the water stress measurement, this 13C, against the deep rooting trait that we've measured. And we can see that there's a strong negative relationship here, which means that in the case that there is more deep rooting, we tend to have less water stress, which is really what we would hope to see, because it suggests that they are taking up water from the deeper soil layers. So this is a very positive result. And yes, that's everything I have to show for today, but I hope it's been an interesting insight into what we can show in the Radimax facility. And again, you'll see more about this in the Radimax video, which is going to follow on from this.
Yes, thank you, Ness. And then I think the program is tight, so we need to go on to Katja Boutin to talk about what they have done at Arvalis in France. So please, Katja.
Yes. Oh, sorry. Yes, hello everybody. I am Katja Boutin from Arvalis, and I will present you our results on SolACE 2020 field trial. So first, yes, I don't really know if everybody know Arvalis. We are running applied research institute on cereals, maize, potatoes, flax, tobacco, and also in forage crops. And we are dealing with research and development on crop physiology, biotechnology, but also crop phenotyping, crop protection, agronomy, and economy and environment for farmers, and feed and food. So we are, yes, financed by growers. So for the experiment in the SolACE project, we manage a field trial in our location in southeast of France, so in UCLouvain. We look at 8 line of durum wheat, and we saw it to have 2 modality. One optimal with well watered and well fertilized, and another one with water and nitrogen deficit. So for each we have 3 replicates. To tell you for the climate of 2020 in this location, it was very rainy in the autumn, so we couldn't sow in an optimal date. So we saw this field trial in the beginning of January, and after that we have a very dry spring. So the field have a drought stress during the elongation of the stem, And then at the end of the development, during grain filling, we have quite a rainy and mild temperature, so quite optimal condition. We had an high impact on yield with these two modality, with two conditions, and also a real effect on genotype. We apply in this field trial our mini rhizotron method. So it's the same method that you have seen for Radimax, but we use 2-meter tubes that we lay in the soil with a specific materials. We put it at 45 degrees in the soil, and then we introduce a camera, a scanner that take a picture at different depths, and in each picture we applied a classified image process. With yes, I will detail it after. For this kind of system, we measure root impact per centimeter squared, like a grid profile. Then we measure length of roots present by unit of soil volume and soil depth, and also average of diameters. Whereas using it for root depth measurement, and also during this trial for to characterize the plasticity of roots to water stress and low fertilizers. So the method is well, we are taking picture in field. We stock them, and then we apply and we assemble each the picture to have a whole profile, and after we apply the segmentation using deep learning reference to detect each root in the picture and in the image, and to separate root for the soil. And at this moment, we could apply two kind of measure. One, it's like the grid impact for a root profile. So in each centimeter squared, you can have a measure of if there a presence of roots or not, and so you have a percentage of roots for each centimeter of depth. And the other one is like to extract physical measure. So we measure the length of roots that are registered in each image, and also diameter of different roots in the image. We extract that in a table, and then we could apply some graphic to analyze the data. So this is the picture of GREU in 2020 with a transparent tube in the soil. And this is the data for each tube. So I present you only two ici, but here, but we had one tube per plot for each variety and each condition. So we measure the impact of density profile, and and you can see that we measured it at 6 different dates, and you can have the progression during the time. And here is the length of the root length density profile, and you can have you can observe a good correlation between the two kind of measure. Then we combine root profile per variety. For each variety, we have a mix. In blue, you have the well water condition, and in orange red, you have the water deficit condition. And for each the 3 replicates, so each replicate was a different plot in the field, so you can see some variation of this kind of measure. And also we have a big progression in 20 days during the spring between the growing period of the stem, and we observe in about root depths at 60 centimeters. We observe in a big progression for the water deficit and low nitrogen condition. We compared deep rooting impact on yield. So here you have a yield, and here's the root length density at 67 centimeters, because it was windows, so it's about 70 centimeters depth, and at this level we could see here the well water condition in the blue point, and in orange the water deficit condition. And you can observe larger root length density at this 70 centimeters depth in stressed condition. And also we have a high variability of genotype, because you can observe here Claudio that that have not developed so much root length in depth condition, but Orjaune I have a very high development, so we can observe this variability in the field. And to be continued, this method for us is very interesting, because non-destructive and useful for deep roots, but not so high throughput. And we have a lot of ways to analyze this data. We are still in progress. And for our SolACE results, yes, this 2020 field trial was applied for very early dry condition, and we tried to we will evaluate again in 2022 a new set of variety selected on their genetic characterization during a SolACE project. Thank you for your attention.
Yes, thank you very much, Katja. And I can see there's one question coming into the chat, and please prepare more questions. We'll have a Q&A after the next presentation. So, but for now, Xavier, Drey from Louvain, who will present work on root modeling in the SolACE project. So please, Xavier.
Thanks, Christian. So good morning to all of you. So what was clearly shown in the previous presentation is that there is a root signal and there are root effects and variation between genotype, but any variable that is measured about roots usually displays a very large variability. We could say that these variables are noisy. They are also complex because we need to describe root systems to touch depth, X, Y dimensions also, plus time, and the environment is changing, the roots are plastic. So it's a really nice playground for people liking to handle complexity. So I just would like to say a few words about modeling to start with. And I will start with a category of model that we call the crop model, because they simulate the functioning of a crop. They don't know really what is a leaf, but they know that there is photosynthesis, that there is transpiration, and these different processes are linked one to another. And so this slide comes from the APSIM model developed in Australia, shows the environmental inputs, so light, temperature, water. And if we move from top to bottom, at the end we have yield, and we see, for example, that light will actually affect through photosynthesis the fraction of light that is intercepted by the leaf. So leaf comes into play. Leaf grows thanks to carbon. Carbon comes from photosynthesis. Since photosynthesis depends also on transpiration, well, this kind of model have the ambition to represent many of those processes at a certain scale. Today, we are more interested into this small area, which is root system architecture, where roots grow to explore the soil and take water from the soil and generate a water supply. And we could do the same with the nitrogen as well. So at the end of the day, these elaborated crop model are kind of a statistical model. They try to make a relationship between yield with the genotype, the variety, the environments, and also the management. And it's a nice tool in that to connect different disciplines together. So phenomics is going to give information about yield, about genotypic effects, about environmental effects. Genetics is describing the genotype. Management and agriculture is describing management as it is. And the function here is the magic function, which represents the functioning of the system with the interaction of genotype with the environment and with the management. Are these models useful? Well, they can be used for a number of different things. One example is here to model can be used to sketch hypotheses about results that we have. And an example comes from the study of maize grain yield evolution from 1920 to 2020. So this is a time series of different varieties, showing that, yeah, the breeders have been doing a good job. But surprisingly, that the genotypes that we have today are more able to exploit space. So if we increase the plant density, so the number of plants per hectare, we see that at low density, the increase has been of less than 1 ton. At medium density, the increase was about 2 tons, but at high density, the increase has been bigger. So one explanation, one hypothesis to that was that actually over this time of selection, without knowing, the breeders have changed the root system architecture from a system that is less deep and more shallow to a system that is more deep and less shallow. And this allows to farmers to grow the crop at a higher density without too much overlap between the root system, and also exploring deeper soil layers. So they tried to put that into equations in the previous model, and they came up with a simulating result, suggesting that actually the genotypes produced in the year 65 would have been actually able to produce more at higher density. It doesn't demonstrate anything. It's just show that the root system effect that is described in this hypothesis would be sufficient to account for this kind of response. We can also use this model to make prediction. And the example here is also related to root depth. One way to achieve root depth is to have genotypes that grow fast vertically. And so by using a simulation approach, François Tardieu and Boris Parent were able to show, again with the same AppSim model, were able to show that the gain of yield we would have comparing a fast and a slow growing genotype would be dependent on the soil depth. So in a shallow soil, having a deep propension to do a deep root doesn't change anything to the crop. If the soil is 1 meter depth, it has a negative effect, and the positive effect will only be achieved if we have a deep soil. It kind of makes sense, but it was nice to show that quantitatively it can work through this complex model. Next to the crop model, we also have different categories, and another set of models is the virtual plant models. So these try to represent the root system with a very fine resolution down to the organ or to the segment level. So what are we able to do with this kind of model? So the example here is a connection of this kind of virtual plant with a soil water transfer model. We are able to take a simple root system to connect the root system to the soil in which it will grow. And this soil will have a certain heterogeneity in terms of water content. And once the two are connected, the model is computing the water potential inside the xylem at every position within the plant. It's also showing the flux of water into the soil from deeper layer to the superficial layers in this case. And it's also giving access to a variable which is very complex to measure, or potentially, well, let's say very complex, is where is the activity actually occurring in the system. And so the classical crop physiologists would rely on soil moisture to say where water is actually taken up, see how the soil moisture is distributed. You see that it's depleted here close to the area where the root system is more dense. But the model predicts that actually water uptake occurs below. So a measurement of soil moisture would not be the right model, the right measurements to evaluate where the roots are active. Once we can also, then I say, use these kind of models to make prediction. And one very useful prediction of this model in terms of deep rooting is that it's good to have roots deep because it gives access to the soil, but it doesn't suffice. You need also to have the right hydraulics. So what is hydraulics that I'm talking about here is two variables that's related to the plant effect. One is what we call the radial hydraulics, which is how water flow from the root surface to the xylem vessels, and it's probably dependent on aquaporins activity and anatomy. And the other one is the axial conductivity, or the conductivity of the xylem. That's how easy water moves along the roots once it's inside. And if we change the ratio of the radial to the axial conductivity from a low value to a large value, we observe, again, it's a simulation, so it's not a true observation. We predict that the distribution of water uptake would be, in this case, rather uniform across the root depth and would be dramatically concentrated to the topsoil in the right situation. So just altering in the two situations, the soil, the root density is represented by the dashed line. So it's the same in this case, but we see that we can have much more water than what suggests the density, or much less. So what we have tried to do in SolACE is to couple a wheat crop growth model called Serious Quality with a 3D root architecture model called ArchiSimple, and with a soil root transfer model called Min3P. So why coupling these three models? So the crop growth model has the ability to predict yield, but it has a very, very simplified representation of the root, which is essentially a root occupancy coefficient, saying that, well, according to the maximum root depth, the relative root water uptake would be linked by a straight line. What the ArchiSimple can do is make a very nice representation of the root with very detailed, very much details, but it doesn't have any representation of canopy, of photosynthesis, of transpiration, or nitrogen used by the plants. What the Min3P model can do is explicitly tell us how water is going to move from the soil to the root, but it has no explicit representation of root architecture nor of canopy.
2-minute warning.
Thanks, I'm nearly done. In the coupled model, the canopy that is inside the crop model is computing the amount of carbon resources. Part of this is transferred to the root system. The root system will be able to grow, to explore a new volume of soil, and also to capture soil resources, water, nitrogen, phosphorus, and so on, that are then back transferred to the canopy. And we then have a kind of a loop with lots of feedback, integrating time, phenology, space, growth, and so on. And one of outputs that we have at this moment with our coupled model is to show, by changing from water high nitrogen to low nitrogen or to water deficit, how this affects soil root density over depth and also over time. And we see that the nitrogen effect will be strong at the flag diff level, but will disappear after. Again, this is simulation, but it can help interpret data. So to conclude, from a general point of view, I try to tell you a bit about modeling, which goes hand in hand with agricultural research. It can give different perspectives on past experiments. It can guide the design of new experiments and make predictions that would contribute to decision making at different levels. So what we are still doing now in SolACE is to fit or to adjust the architectural model and the crop model to the 500 genotypes that we had in the SolACE project, with then the aim to analyze by simulation how above traits and below ground traits value in a wide range of environmental scenarios. Thank you very much.